Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond.
Welcome to today's episode. Today's topic is From Letters to Physical Blobs, Visualizing and Studying RNA, the Ultimate Mediator of Life. To help with this topic, we're joined by Saron Tekie, who carried out her bachelor's in biomedical sciences at the University of Birmingham, followed by a master's in cancer and molecular biology. Saron is now carrying out a PhD in evolutionary genetics in UCL with Gemma Murry. Welcome to the podcast, Saron.
Thank you. Thanks for having me.
It's a pleasure to have you on. To get us started, I'm really interested in this topic of RNA and how it's the ultimate mediator of life. Can you tell us a little bit about the project that you did with Konstantin Reder at King's College?
Yeah, so I'm glad to be back here. Yeah, so I actually did this work downstairs on the third floor at the Randall with Konstantin Reder, like you said. And we just were able to model a 48 nucleotide RNA, which is a collagen mRNA. So it's directly translated into the protein, right? And we modeled 4 mutants. So one was wild type, and then 3 mutations, which had either a 1-point mutation or a 2-point mutation or a 3-point mutation. And the motive for this was we wanted to see what structural differences these mutations made in the RNA structure. And we also had a complementary protein, which is involved in binding to these RNAs, well, to the wild-type RNA. And upon binding, what happens is the collagen mRNA, which is what we're modeling, is upregulated. So it's translated at a higher rate. Mm-hmm. So this is something that happens in a lot of fibrotic diseases, which means there's an overexpression of collagen, not only in fibrotic diseases, but also this is a ubiquitous process in inflammation. Mm-hmm. So in inflammation, the collagen expression is upregulated, which can lead to things like dysregulation of the tissue and just obstruction, right, of the function. And so we wanted to know how this RNA binds to its complementary protein, which in this case was LOB6. And so the first part of the project was just modeling the structures of 4 mutants, one of which was not a mutant, is the wild type. But and this was done using molecular dynamics, which I can go into more.
Yeah, amazing, really interesting.
Just to clarify, when you have dysregulation of translation or protein production, you have the same amount of mRNA, but you're just making more protein?
So we have the RNA, right? And its regular function is that it will bind to a protein called LOB6, and once this binding occurs, the RNA is translated at a higher rate, so it's overexpressed.
Okay.
So you have an overexpression of collagen when this RNA and this protein we're talking about bind. Mm-hmm. And you don't want that, because when collagen is overexpressed, it's like scar tissue, mm-hmm. right? And it, for example, can obstruct the tissue, like the function that's supposed to be happening in the tissue. So we want to understand exactly how this binding occurs between our RNA of interest and our protein of interest. And the way to do that is to study the structure of the RNA. And the reason why simulations are really useful in this process is because RNA is very dynamic, and it doesn't just have one shape when it binds to this protein of interest. And these regions are called intrinsic disordered regions. They don't have just one 3D conformation which binds to its target. They're very dynamic. So if you have a simulation which is basically a movie. It shows you what all the different structures adopted are by the RNA. You can see exactly how it's behaving over time and not just one snapshot of the structure as you would get in, for example, crystallography or crystallization.
So how do you go about first finding out the different components and structure of the RNA if it's so dynamic? Like, do you use traditional tools and techniques like NMR, or are there different techniques that you have to use when it's such a intrinsically disordered protein? Or
that's a good question. So we do use NMR to determine the structure of the proteins, for example. Okay, so in this project, what we did was we have a sequence of the RNA, right? And then from that, we predict the secondary structure. And there are softwares to do that. And then from the secondary, you predict the tertiary structure. So a secondary structure is just the sequence of nucleotides in a chain. The tertiary structure is a 3D conformation of the chain of nucleic acids in the RNA. Yeah, the difference between these is, if we go back to A levels, is the kinds of bonds that are present. So in the secondary structure, we would have things like hydrogen bonds, van der Waals forces, and things like that. In a tertiary structure, you might start forming like disulfide bonds, which is basically they create a 3D shape instead of alpha sheets.
Yes.
And beta fold.
Right. So once you have the tertiary structure, it is basically represented in a coordinates system. Before we simulated, we just have a tertiary structure represented in a coordinate system. So one atom, you can think of it as a ball, and then the bonds between two atoms is like a line, right? And this is classical MD, molecular dynamics, in which every atom is represented by like one data point or one ball, let's say. So this is where we start before we get into the simulations.
But if the same sequence of amino acids and nucleotides leads to different 3D conformational structures, then what do you end up? Do you end up with 20 possible different conformational structures based on the same amino acid sequence that you then go and model? Or do you just pick your favorite one?
Okay, so this is mRNA, right?
Okay.
We're starting with a sequence of nucleotides, which is the mRNA. From that, we predict the most likely secondary structure of the mRNA, and then from that secondary structure, we predict the most likely tertiary structure. So, yes, there could be different conformations possible. There are different conformations that are possible, but we pick the most likely one based on the predictions that we get from the softwares. For example, if you're interested in specifics, RNA composer and RNA fold, they're used to predict secondary and tertiary structure. Yeah, so that's what we start with in our pipeline of simulation.
And the probabilities of these different structures, is that based purely on the chemistry? Is it based purely on which nucleotides
are likely to bond with which nucleotides? Yes. And these algorithms are sophisticated, and they predict most likely on the forces which conformation is most stable and most likely to be held by the particular sequence of nucleotides in the mRNA.
Is RNA fold, does it have anything to do with alpha fold?
I don't know why the naming is similar, but RNA fold is for nucleic acids. Alpha fold is for proteins.
Would you
right?
Would you say that potentially with tools like AlphaFold and other dynamic tools that exist for proteins, do you think that they have a much higher accuracy percentage than tools for RNA, just given the fact that it's more disordered?
Is it more disordered though?
It can have more, it can bind to different things, and it can have
Yeah.
different conformations.
That's true, yeah. That's a good point. Yeah, I would say we might be more confident with AlphaFold, but I haven't really explored AlphaFold like that. So,
if in the context of your research, your goal was to find out how this RNA was binding with this particular protein,
yeah.
would you do most of the modeling with the dynamics of that specific protein in mind, given that the conformation is somewhat dependent on what it's binding to?
Yeah.
Yeah.
You could model both the RNA and the protein in the same space, but in this project we modeled the wild-type RNA and then 3 of its mutants.
Okay.
And the reason we know that the RNA binds to the protein from past studies, but also we're really interested in this RNA because it has a very specific site which we know is the section binding to our protein. So we altered specific nucleotides within that specific site, which we call the bulge. So there's like a little bulge inside the nucleotide sequence, and it's basically the active site, let's call it, which is recognized by the protein. So in this case we didn't model the protein computationally like the RNA, but I did make the protein to functionally validate later on.
Cool.
So yeah.
Nice.
So going back to what Carolina asked about techniques used to look at the RNA structure, you mentioned that NMR is one that is used, but are there other methods used besides the computational modeling to determine the RNA structure?
So the NMR is a very, very detailed, and it can show you where every atom of a structure is, and we do use that as a checkpoint, as a reference. For example, if you want to understand how a particular protein or RNA binds to another protein or RNA, it's nice to have a definite scan using an NMR. But in terms of RNA structure, it's a challenge because of how dynamic they are. So for example, if you want to use X-ray crystallography, it obviously crystallizes it, so it's it's a frozen one shape, but that doesn't mean that that's the only shape that the RNA has, or it's the most effective shape for the function. It's just a shape, and so it doesn't really capture the whole picture of the RNA structure that's involved in the function that we're interested in, which is why simulations over a period of time can capture more information about the structure of the RNA. In terms of other methods past the X-ray crystallography and the simulations and NMR, I am not sure exactly I don't know, there could be. I don't think so. I didn't really get into that. That's curious. I don't know.
But so do we know that they are dynamic because of simulations? Because if there isn't a way to test it directly, then how do we know?
Well, I think we know because we find lots of different conformations.
Okay.
So
in frozen time,
yes,
you can still find many.
Yeah, I think so. That's what the literature says, right? So
Yeah.
I know that they're dynamic, and we have disordered regions where we can't really determine this is always the shape that they hold. So it must be that we've seen that they can hold different shapes with the methods that we have. So NMR kind of addresses that problem with crystallography as well, because it doesn't snapshot like X-ray crystallography does for RNA. Cryo-electron microscopy is also used to determine structures.
Wow, I didn't know cryo-EM could go down to the RNA level. People use cryo-EM in neuroscience, right? And then we look at what do you use things that are much bigger.
You can see ion channels.
Yeah. Well, cryo-EM is on the level of X-ray crystallography. So that's what it's trying to say. Yeah.
I'm just not thought below the protein level, I think, in terms of structure.
Yeah.
But this is super cool.
Yeah.
Wait, so before Before we go into what you found, the results, can you explain a bit we've kind of touched on it already, but can you explain the importance of RNA structure?
Yeah, like a lot of things in the cell, structure is what determines the function of the RNA. If it, like the classic lock and key hypothesis, if the structure is not complementary to whatever it's binding to, it's not going to bind, and then it's not going to initiate any downstream signals or whatever it does, whatever the function is. So this is literally what we study in order to find therapeutics or design drugs to address problems of, when the shape is not complementary to whatever it needs to bind to. Which is why we need to study the structure as accurately as we can, and we're implementing techniques like molecular dynamics to capture more accurately the structure of RNA.
And that's how it goes from being letters to blobs.
Exactly. Now it's not letters, it's blobs, and you can see it and play with it.
I think it's particularly interesting because we always think about protein structure.
Yeah.
But we don't think about what comes before the protein, which is the RNA, and the fact that will also determine whether or not the protein is made in the first place.
Exactly.
Yeah.
So is it more important? Well,
No, go on. Throw in a hypothetical.
They're both important. The chicken and the egg, they're both
because
required.
also the RNA is binding to proteins, isn't it?
Yes,
so
And
exactly.
it creates the own proteins that it needs to have its own structure.
Yeah.
It is chicken and egg.
Yes, it's hard.
Do you have a favorite? Do you have an opinion?
I mean, I think RNA is pretty cool. Is once you have the protein, unless the protein is needed to bind to another protein which does the function. I mean, it's hard. You need both, but I'm going to say RNA on this occasion.
Is it because of the dynamics? Do you find them more interesting?
Yeah, I think so. I think it's maybe it's the first step past the DNA that kind of starts this whole process of the production. So, it's really, they're all important, but I think RNA is interesting in that sense. It's in the middle of communicating that message.
The mediator.
Yes, exactly.
Of life.
Of life. Yeah, it's pretty cool.
So going back to your collagen study and your 4 mutants of the RNA and how they bind, what did you find?
Okay, so it was a pretty cool project. So we know that the wild type binds from previous studies, and I also validated that it binds. But what happened was, when one of the nucleotides was mutated, so it's a 1-point mutation in the central region, which we know is the part that's actively binding to our protein. The bulge, exactly. The binding function was lost. So having this one nucleotide change inside a central area basically has lost the function. And so this study was comparative in a way where we can then look at the simulations and say, okay, the wild type binds really well, really clearly. One nucleotide change has lost the function. So what's the difference in the simulations of the wild type and the first mutant, which is only a 1-point mutation? And this is where you get into analyzing the trajectory, which is just a movie, right? It's a movie, it's a simulation over time. So you analyze the movie to see what's the difference between the two, right? And interestingly, the structures of the wild type and B1, let's call it, B1 is the 1-point mutation, were not that different. So you can kind of see how it's very sensitive and intricate, because the overall tertiary structure wasn't that different, but the only difference that you could see was that B1, the mutant, was more dense. Like, the structure itself was more dense. And it was shorter in length. So it's just you set up different scenarios, right? And then you test for function, and then you see there's a difference in function. So then you have a look at your simulations and say, okay, what's different here? What's different in the structure that we can see that has resulted in this difference in function, right? That is of interest to us. We know that mutating just this one nucleotide now has lost the binding between collagen mRNA and its regulatory protein LARP6, right? So that's of interest to us because we're interested in basically reducing overexpression of collagen, which is found in fibrosis, for example. So we can go from that. So that's our bigger interest. Okay, maybe then we can think about small molecule design that can address the binding between the two to kind of lower the overexpression of collagen, and therefore play a role in therapeutics.
And what was your readout of the function? Did you actually look at collagen protein levels, or did you just look at the direct binding that was happening between the RNA and LARP6?
Yeah, so we said the first component was simulating the RNA structure, right? And then the second component was validation of function. And in this validation, I used a technique called isothermal calorimetry. So it looks at binding.
Okay.
So obviously, to have the protein, I needed to make the protein, and I expressed this protein in bacteria. So you express it, you purify it, and then you have the protein. It wasn't that quick or that simple.
Yeah.
But
it never is.
but we made the protein, and we have the RNA, right? So we put them in the machine, and it's a machine that's very sensitive to changes in heat and enthalpy. So anytime there's a reaction or a binding, you can think, it's going to release heat. So that's how you kind of measure the event of binding.
Yeah.
Right. So it's not the protein levels.
Yeah.
It's the occurrence of binding.
Yeah.
Whether there is binding or not.
That's super cool.
Yeah.
Yeah. It is. Yeah. It was a really useful tool. It was great. So we just have wild type binds perfectly. Well, we have a very characteristic sigmoid curve to show us that there's binding. From that we can calculate KD and other values that can kind of tell us a little bit about the nature of the binding, how well it binds. And then, for example, for B1, which is the mutant, the 1-point mutation mutant, shows no binding at all. So it's like a flat line, which is pretty cool. So from there, we went on to basically design a novel mutant, which we were like, okay, what happens if we change, for example, these nucleotides somewhere in an interesting region of the RNA? We hypothesized that doing that should not be as detrimental as changing the one nucleotide, but it shouldn't bind as well as the wild type.
Like an in-between.
Yeah, like an intermediate. And that can that is a great source of information, because you can it's like hot and cold, right? You're close, you're far, you know that game? When you're...
Like Marco Polo?
Oh, yeah.
Yeah, so you can kind of use it as a mapping kind of system to track what's beneficial, what's not beneficial to the function in terms of structural change. And it worked really well. We weren't expecting it to, we weren't super confident that it would work, but yeah, we changed a couple of nucleotides somewhere else in the structure.
Not the binding site.
Partly in the binding site, partly just the outside.
Okay.
And I mean, it's much easier to see this, drawn out. But so there, and then
it's funny that keeps happening in all our biochemistry.
Yeah. Biochemistry is so visual.
Yeah. So this intermediate that we designed showed some binding. I think this was the most interesting part of the project, is that we can actively manipulate the binding from the structure, from changing the structure, and that was great. And we have an intermediate binder a non-binder, I'm just making up these terms, and a binder.
Yeah.
So we can kind of, now we know what the functions are, we can go back to the structures and think about, okay, what's the difference between these structures that has led to this difference in function? And yeah, having that intermediate takes us from just a binary, this binds, this doesn't bind, to how could it be binding? What is important? For example, is it the size of the bulge? Is it the length of the structure? Is it, what conformation is more beneficial to the binding? And yeah, so we take these kind of things into consideration.
So did the intermediate binder have an intermediate structure?
Yes.
Okay.
In a way. In a way, yeah, it did.
In terms of in the same way that you mentioned the non-binder was shorter?
Yes.
So
From
was
end
it
to
again?
end,
Yeah.
it was shorter, but everything else looked incredibly similar to the wild type, which was very interesting because, characteristic features of the wild type that we thought were important, like the central bulge and there's a little loop at the end, were all present in the B1. But so the only big difference was the end-to-end length of the B1 mutant, which has lost this, which made it lose the function altogether. So it kind of maybe tells us what's important in this binding interaction.
So did the intermediate binder have an intermediate length?
So the intermediate binder had a similar length to the wild type. So the difference between the intermediate and the wild type was that in the intermediate, the central bulge was bigger than the wild type. I mean, there was a lot of differences that we can go into. So we can measure things like the size of the bulge, right, in the central. There's another structure called the stem loop. We can measure how big that is and whether that's always present. We know from the past investigations that is important and is always present in all our mutants. We can measure the stability of the structure. I think a classic measure in MD is RMSD, root mean square deviation. So it's kind of, it gives you a measure of how stable the structure is it fluctuating, and there's lots of parameters that you can look at, but I think the main point was we see a difference in the function. We want to see what's the difference in the structure.
Could you tell us a little bit about what the function of these different structural bits are, like the bulge and the stem loop?
Yeah, so this binding between the RNA and the protein is sequence specific. So what's being recognized is a sequence on the mRNA. And this sequence specific binding is basically the central bulge, where you have like 5 really important nucleotides which are needed for the protein to bind. Yeah, so it's basically, it serves as a recognition domain, I guess.
And the stem loop?
That's a structure that's predicted to be very stable, and it's a common thing. We have RNAs with stem loops. Like RNAs some of them tend to take a stem loop kind of structure. They tend to have stem loops in them. So it's one of like the common conformations of RNA sequences.
And so then how would you go about finding therapeutics for this? Like you mentioned either introducing a protein or would you do some more mutations to...
Yeah. So going back to the Marco Polo analogy, the more you do, obviously you'll be led by your previous findings, so you can kind of hone into more informative kind of experiments. But yeah, so in the future, taking this project, you would look into what kind of molecules would be good interventions. So you know that if you do more like experiments, you'll know what kind of structures are helpful to the binding and what kind of structures are not helpful. So you can kind of expand on that and design small molecules or sequences of RNA or other nucleotides to kind of impede or stop that binding. So you would have to simulate other small molecules in this scenario as well. And one of the future directions that we had was also we could simulate the protein as well. so that we can see the binding in simulation. So that would have been the next steps for this project.
Do you have any idea how you would administer these molecules? Yeah, these drugs, if it was like either an animal model or something? Yeah.
Obviously, you would have to go through the whole, you're not going to stick it in people straight away. You would have to go through the regular,
but would you consider...
In vivo, in vitro.
Yeah, of course. But would you consider this like gene therapy? Like, how would you class it?
I think it would just be...
Because you're changing, the nucleotide sequence, right? You're affecting...
That's interesting. I think you would... if we're designing small molecules... I honestly don't know. It's so early. It's so early. I don't know what's the best way to... It's so early. So first we would understand how the binding is happening. Like I said, they're so dynamic right now. Like, how do we go about designing molecules for RNA therapy? Yeah, that would take, I think, a lot of simulations before.
No, of course. I was just curious because it's like in between, the protein and the DNA world, And
actually, I mean, like you said, if you could target the protein. So if you know what RNA is binding to the protein, maybe you could design something similar to the B1 mutant that could bind to the protein.
Okay,
so that could basically cut off that that binding.
Cool.
But I'd have to think about that. I don't know.
Just curious.
I mean, I guess it really helps actually that the structure is so related to the function, because then structure is relatively easy to change. Like, you can't immediately... Like, if there wasn't a direct relationship between the two, if the structure wasn't predictable by a single nucleotide, then you wouldn't be able to change that. So it's pretty amazing that you actually have that direct relationship.
I mean, it's a very interesting observation to lose function just because of one nucleotide, and the overall structure doesn't look like it's not completely lost just because you had that one mutation. So it's a very interesting observation, and to have an intermediate, it's even more interesting because you can kind of study how the binding is happening. It's not just that it's lost, but what characteristics determine successful binding between the RNA and protein. So I think it's a great way to study structures, and I don't know if we were lucky or it just worked, but having that intermediate, I think, was really, really helpful.
That's really cool. So when you're modeling all of these RNAs, you also talked about simulations.
It is.
How do you simulate them?
So it is modeling, and the way you model these structures is you like I said, we start with the secondary and tertiary structures, right? And create a coordinate system of these atoms where they're located in 3D space, and then we have softwares, should I say, or tools like Amber, for example, which I think is so cool, so exciting. People should check it out because they have lots of helpful tutorials that you can explore yourself, and you can do this. I mean, you probably need a lot of computational power, but it's really interesting regardless. You can have a look at how Amber works, but so you just have an input of the coordinates, and then you minimize them, because sometimes these predictions can give you unrealistic conformations a little bit, and that can, where the energy is too high in some sections or some bonds, or so you want to minimize the whole structure to get it to a local minima where it's most stable, basically. And then from there you can heat it up to a temperature that you want to heat it up to, and then you equilibrate the pressure, and then you can start the simulation. Over time they can kind of move in ways that they do according to the force fields and everything that, according to the force fields.
That's really cool, because I've never thought about the fact that like DNA, RNA, protein dynamics would be different if we weren't on Earth.
Yeah, exactly. So
Yeah. So you could also simulate it based on like other planetary forces, no?
Yeah.
Technically.
I guess. Yeah.
I wonder if people in astrobiology use these sorts of
Probably.
tools. Yeah.
I wonder if they would have to use different force fields. So,
Yeah.
for example, in tools like AMBER, you would have like a DNA force field where basically it's an algorithm that would simulate the forces you would expect these atoms in nucleic acids or DNA or RNA to be in. And I mean, these models are so incredibly sophisticated, I wouldn't be getting too much into them, but yeah, it's pretty amazing. So we just want to, basically, it's modeling, right? We want to create an environment that the RNA would be in naturally. So you would have water in there, ions, you would balance the charge, and you would have obviously the forces that would be acting on the atoms. Yeah, so if you think about it, it's just like how you would model anything else. So if I told you to build a model of
something non-biological, obviously I'm just
something
thinking of
non-biological.
neurons. Like a building?
Yeah, like you would think about
Its location.
Yeah, its location, what else is around it,
gravity.
Yeah. So I mean, I think it's pretty cool. So the shape that I had the RNA in is like something I could get as close to a sphere. So it was like an octagon, like octagonal sphere, let's say. So yeah, it's just like a bubble of water and ions and the RNA inside. And you just, once you applied all the conditions that you think you would be in, obviously with the force fields and the ions and the charges and the water, then you just see what it does over time. And that is what we call a simulation.
Yeah.
Or the trajectory.
Yeah, it's really cool.
Yeah.
What sort of timescales do you look at?
So we're talking picoseconds.
Oh wow.
Which is actually important because sometimes that time period is not long enough for the RNA to adopt certain, might not be long enough for the RNA to morph into whatever shape it needs to. So we take that into consideration in relation to the time scale of the reactions. So if the reactions take a lot longer, we might not see the structures that are needed for the reaction to happen within our simulation. So we do have to take that into consideration, but this is something that takes a lot of power, and we're looking at picoseconds. Well, 200,000 picoseconds, but still, picoseconds. So they're very quick.
Yeah.
Yeah, there's probably like 1 million picoseconds in that snap.
Well, yeah, probably. Yeah, I think it's pretty interesting in that it's I don't know, I think it's a fun journey to be able to go one step to the next step, to the next step, and then you have a bubble of water and other things with RNA in it.
Yeah.
And you can make a shape which binds to another shape.
That is so
which
cool.
keeps you alive.
Yeah.
Well, in some cases.
Yeah.
So, yeah, I think it's pretty interesting.
Nice. Thank you for explaining to us the journey of letters to little blobs and how RNA mediates life.
Oh, no worries.
Before we finish, I also just wanted to ask you about your PhD project and how that's going with evolutionary genetics.
It's going pretty well. It's a new kind of field for me, but it's something that I'm really interested in. So this would be molecular evolution, and I'm interested in how these sequences are changing, evolving in relation to, you know, the environment that they exist in. And obviously antimicrobial resistance is a big problem, and it's only going to get bigger from here. So yeah, it's really exciting. I'm learning a lot of bioinformatics, and interestingly enough, it's been so fun for me to see the sequences and understand, oh, this is that protein, right? It's got a start codon and an end codon, and I kind of relate it to a lot of times to the structures that would be formed from it.
Yeah.
Because I just really enjoy looking at the functions that come from the structure. So it's been really helpful to have this background in my current genetics project. So yeah, I've been having fun exploring antimicrobial resistance and resistance proteins, resistance genes, and how they might be changing, how they might be being transferred between different populations. So learning a lot and having fun.
Nice, exciting.
Yeah.
I see kind of the similarities, like they are very different.
I'm
but
very happy to hear that, because I'm constantly trying to, I mean, it's very similar to me, and I'm interested, I see the connection between them, so I'm happy to hear that.
And also the way you described it, it makes it clear that it's like, it is a language, how proteins are coded in these letters.
Yeah, it's pretty. So all I do is I see letters
all day.
Nowadays, all day. Lots of letters, and it's always such a nice feeling when they make sense. And then they're more than letters, and you can kind of make interpretations that are can be so impactful. And I don't want to sound cliche, but they're in all of us. That's how we function. They're everywhere, and it's kind of really cool to see that.
Yeah. Super cool. With that, I think we'll end today's episode. Thank you so much for joining us today, Saron. It's been a pleasure.
Yeah, thanks for having me. It's been great.
Thank you so much for your insights into really going into the depth of the layers of DNA to mRNA to protein.
Yeah, no, it's been fun.
We hope you enjoyed today's episode. Please feel free to share it with your friends. Our Instagram handle is @neuroverse_pod, as well as our Twitter handle. Check out our website for blog posts and resources on today's episode, neuroversepod.com. We'd like to thank the Biochemical Society for supporting Neuroverse, a podcast about science and philosophy. This episode is part of our series to disseminate research done in biochemistry.